And greetings, everyone. Dr. Mark Kelly here. We are going to get into chapter 14, special senses. Now you say, what's special about special senses? Well, not necessarily anything, except they are how we perceive the world. Okay. And what you think are the special senses is probably not correct. Okay. And the all but one you probably guessed correctly. And tell you the truth, it's right in this location and including there. So what were these? Sight, sound, smell. So there's three S's right there. Of course, taste. Okay, so there's four of them right off the bat, right in our little area. But what's a fifth sense or something? And people would say touch. No, it's actually equilibrium, or our ability to balance ourselves and to know where our body is in space and motion. So that's the special senses, and let's get started into it. Oops, wrong screen. Sorry. Let me get back to here. Stop sharing to here. And now it should be coming up. Very good. And we'll go big. All right. So, what are the special senses? First, I'm going to talk about how the transition takes place from something on the outside to how we perceive it on the inside. And I'll also later go into how that's not always correct. Okay. But anyway, so this is one of the first areas. Let me pull this over a bit. And um, this was just an example of, us seeing, when temperatures are high, sensing skin, and reflexive withdrawals initiated by the muscles of the arm. This is, the sensory neurons are activated, and this reflex is done without conscious thought. Our bodies protect itself. And I put this in, of course, no matter how stupid we are. So in other words, if you touched a burner like that, obviously your skin would get fried, literally immediately, but you would actually pull your hand away before you knew how hot it really was. Isn't that kind of interesting? And we're talking split seconds here now, milliseconds, but still, that's a reflex that we've built into our bodies to protect us, say, from dying or walking into a fire. Anyway, here's the main senses we were just talking about. So vision, known as visual. Smell, go ahead and know, is called olfactory. Hearing, auditory. So that makes sense too. Taste is gustatory. So that's kind of an unusual word you probably haven't heard of so much. So gustatory. And then we have equilibrium again, which is balance and knowing where our body is. Okay. So what's the receptor for vision? The photoreceptor. It only detects light. Smell and taste are both chemoreceptors, but smell is airborne and taste is either liquid or solid. So that's the big difference there. Hearing, what are we actually hearing? Vibrations. Interestingly, we're hearing vibrations, and that's a type of mechanoreceptor. Now, a lot of these receptors we'll find out are named hair cells, and this one in particular is one, but that's sort of almost a generic word because but when we refer to hair cells, we're going to go ahead and stay with the hearing part of it. And again, taste is a chemoreceptor, and of course, equilibrium is three different types of things, but the vestibular apparatus is part of it, and the proprioception is part of it as well. And vision is part of it, but it helps with our equilibrium. Okay, so again, rods and cones would be for visual. Olfactory sensory neuron would be for the olfactory. Of course, hair cells, again, with the cochlea, are for hearing. And there's actually something called taste receptors or gustatory receptors for taste. And they can pick out, of course, the five main tastes. And then there's some other discrimination we can make. And then vestibular apparatus, frontal and cerebellum. So And it's not transitioning here. 1 second. There it goes. Oh, that was odd. So there's different types of receptors, obviously, for different things, but focus first on this right side of the screen where an external stimulus is picked up by something known as an exteroceptor. So go ahead and know that word, exteroceptor. That's again something from the outside world. If we were to detect our own blood pressure or our own, say, blood gases, chemicals, that would be an interoreceptor. But it's specific for that stimulus. So remember, we just said, how do we pick up light? Visual, photoreceptor. That's all it can pick up. It then will translate that into a neural signal. So whatever we see from the receptor has to be now transitioned into a neural signal so it can be communicated to our brain. Okay, but that neural transmission is going to be coded somehow. So it'd be like, say it was Morse code, you were trying to do dot, dot, dash, dot, dot, dash, and you were saying SOS. Well, that would be, a way to transition a feeling into a signal. And then finally, a brain perception. So it's actually what we actually perceive from that neural signal. And that is, where interpretation can take place during actually both of these stages. But yeah, it's a portion of our brain that actually can decipher that signal and make sense of it to give us what our perception was. So these are just three examples of different types of receptors. Free nerve endings, we'll get into this with this one. Encapsulated endings, this is what photoreceptors in eyes. Specialized receptor cells, they're neurotransmitters, and they would be bipolar cells, which again are kind of rare, but these are things. So anyway, those are just three kinds of receptors with their neurons. Okay, the tongue would be one example of gustatory. So it is what picks up our tastes. Now, what's interesting about taste is that it can be interpreted, you could say, because of the contribution of other senses. And I'll get into that in probably the next slide here or so. But the tongue is covered with small bumps called papilla. So go ahead and know a papilla is what's actually containing the taste buds. So I could ask a test question. What is actually we'll call it the organ, the part of the organ that's picking up the taste sensation, and it would be the papilla. So they're sensitive to those chemicals. And again, it's gustatory receptor cells that actually would then interpret what taste that was. Now, one time they used to think that we had certain taste receptors in different parts of our tongue. That's not the case. We actually have all the receptors in all parts. But all right, let's keep going. And again, it's chemicals dissolved in saliva. That's what we taste. Okay. And okay, we're moving on to the olfactory. So hopefully we'll come back to what I was going to tell you. The olfactory system, of course, smells. If we were to smell coffee and then taste it, we would get the overall sensation of what that quote unquote flavor was or what it was. And so smell is definitely a huge portion of how we perceive how something tastes or its flavor. So the olfactory neurons in this case pick up those dissolved, okay, in the air, and then we'll interpret it. Believe it or not, when you smell poop or a fart, you're actually having small bits of that odorant in your nose. Yeah, it's kind of gross, but we're talking molecules. So anyway, yeah, it's in the ethmoid area of our head. So if you remember back from anatomy and the skull, the ethmoid was basically your nose area. And yeah, so we're going to pick up, olfactory sensations through this olfactory bulb. And it's very well connected with emotions and memory, as we already discussed. So, and they're just going through this. I'm not going to make you know this, but again, these specialized types of neurons. These bipolar ones are going to be um, in there, and they're just showing a slide of that. Okay, the pathway for hearing. What are we actually doing here? It's a vibration again. So we'll get into this more, but whatever we hear has both pitch and volume. So we're going to hear a certain pitch, certain volume, and then we interpret those vibrations as sounds. And we'll get into this in our lab. But structure of the year. I think I'll go through it on another slide here, but there is a pathway that this sound travels through. See if it's on this. Yep, good. It is. So this is a slide I was hoping to find it on. So the auricle is, and you need to know this in order. The auricle, our outer ear, our ear basically, but what you could see is the auricle. It then goes through your ear, what you call ear canal, but the auditory canal. So it's going to make its way down into that auditory canal. It then hits this tympanic membrane, aka the eardrum. So the eardrum is the tympanic membrane. This thing gets translated, you could say, but transduced into the vibrations by these ossicles. They also amplify the sound, so they can amplify the sound, much bigger to where the rest of the system can now pick it up. But again, it's a wavelengths that are going to change how this sound is transmitted, and that's again with pitch. It'll then go into the oval window, which is right there. Oh, I'm sorry, right here. It's going to go into the cochlea from there. So whenever you hear vestibular cochlear, no, the cochlea is the hearing part. The vestibular apparatus is actually the balance part, or knowing where our head is in position. And then finally, it's going to come out the round window. So go ahead and know oval window is before a round window. And that's the pathway of hearing. And you don't have to know the particular ossicles or the order of them. Okay, and they're just showing here in more detail. I'm not going to hold you to this, but they're just kind of showing right there in your ear is where these different parts are. Scala vestibuli, of course, vestibular would be type. Scala tympani, like tympanic membrane, more of the hearing. And then there's this cochlear duct and tectorial membrane. So this is be all part of this, well, there's the organ of Corti even. So it's all part of this larger structure. But I'm not going to make you know these particular things. And the hair cells again. So like I said, there's hair cells for everything, but in this case, there's stereocilia to actually detect where our head is in motion, and the hair cells will be right here, but to also pick up the sound. So hair cells, mechanoreceptor with an array of stereocilia. And again, we'll show you how that can translate head position in a bit. And yeah, anyway, it's in equilibrium with our gravity. And okay, so how do we hear sounds? We do two interpretations of it. Then this is based on what they call the place theory of pitch detection. So frequency, how many waves are, the frequency of the wave. So how many waves per, say, second? Okay, that would be known as hertz. That is pitch. So if you hear it of FM radio, it's frequency modulation. That's for pitch. The size of the wave is going to be the amplitude of it. That's volume. So AM radio would be more volume. So go ahead and keep those two things straight. And again, volume and pitch would determine. So high frequency pitches would be, of course, the high sounds. And that's, like, say, 20,000 hertz. And then low frequency. Can other animals hear higher or lower than us? Yes. And of course, there's some animals that are very dependent on their hearing, aka bats. They actually use echolocation, but because they're blind as a bat. All right, so let's keep going. And this is what we were talking about, the stereocilia and how it can interpret head position. And this is why you couldn't stay balanced when you were spinning your heads um, for our exercise in class. Oh yeah. Go ahead and know that the saccule, like a sack is heavy, that's up and down position of your head, while the utricle is the horizontal positioning, and rotational positioning is in the semicircular canal. So again, saccule, vertical, utricle, horizontal, rotational or circular would be the semicircular canals. And here's that rotational stuff we were just talking about. But some people like the spinny rides at amusement parks. I no longer really do. Like I said, I used to like them. Now I'm more barf. Actually, I don't think I barf at one, but at the same time, I'm not a big fan of the spinny rides like I used to be. All right, keep going. Oh, and your eyes. Yeah. So believe it or not, if you spun around a whole lot and then look, stopped and then try to look at a person, your eyes would actually be going back and forth, and that's called saccadic movement. And we'll recreate that in class in the lab. But our um, eye muscles are actually associated with different sensations outside, so we can actually focus in on a given target because we move our eyes in association with how our head is moving. So it's all coordinated. Okay. There are a lot of structures of the eye that we'll need to know. I want to go through some now relatively slowly here, but the outer part of this part of the eye. Okay, so this part of the conjunctiva. So if you've heard of conjunctivitis, that's a this is the cornea right here. So the cornea is just underneath it, and they're not showing, yeah, the anterior cavity here. So this is known as the aqueous humor, and again, the anterior chamber. Then you have and then if you were to look at the eyeball, the pupil is in the middle. That's always the black part. The iris is what's actually closing, open and closing. So people call you have a constricted pupil or a dilated pupil. Pupil's not doing anything. It's the iris that's going in or out, and they're using ciliary muscles to do it. But that would be the colored part of your eye. So that's what gives you color. And the sclera is the white part of your eye, and that's shown actually in other regions out here. But the sclera is the white in your eye. Okay, so again, you had the cornea on the outside, this anterior chamber. This lens is what actually bends the light. So the lens is important here because um, it's what's responsible for the focus in our vision. So it's either bending to send when the signal is very close up, or it has to relax when something's far away. And that ability to take in both is called accommodation. Accommodation. Anyway, after that, we have the large vitreous humor. It's a gelatin-like substance, and this takes up most of the eyeball. In the very back part, we have the retina. So they're showing it right here, the retina. There's a central part of the retina with the very high density of cones. So we have our best color vision in the fovea centralis. So it's called the fovea centralis. If you've heard of the disease macular degeneration, they sometimes call it the fovea macula, but it's part of the retina. But it's the fovea macula would be part of that. So within this region, there is an optic disc, which is your blind spot because the optic nerve needs to go through that. So the optic disc, again, it's the blind spot because there's no rods or cones there. And because, again, the nerve has to make its way through to transmit the signal that you said. But the retina is basically our film screen. So the lens will try to bend light so it hits the retina with proper focus. If it doesn't, you need correction, like I have, and like anyone that wears glasses has. So you need some sort of correction so the bend is correct for that particular image, like the length of that, where that image is. We'll get into that a little bit more. Anyway, get to know a lot of parts of the eye. Okay, photoreceptors, not so concerned you know about all these, except rods are for low light or highlight, so the amount of light. Basically, you call it black and white vision. Cones, color. The cones are color. And we have three kinds of cones I'll show you in a second. And here would be an example of rhodopsin. So they're in our rods. But it's actually kind of interesting that we have chemicals that transmit this. And so the light will change these chemicals, which then that. So if we had a really bright light and we get that after image effect, it's because the chemicals are still existing there. So the receptors are still picking up that stimuli because it was strong enough. But a photon interacts and it alters these chemicals, and then we get perception of light. And again, retinal is one of the things. So the retina picks up this retinal, which is also known vitamin A. Okay, so this is the thing on color blindness. And pretty interestingly, it's where we have three cones, three sets of cones, or we call it types of cones: blue, green, and red. And you say, well, why don't we have yellow instead of green? Well, we don't. Okay, but because those are the primary colors in blood. Well, this is just the way it is. And in males, of course, have a much higher capacity to have color blindness because it's on the X gene. If males only have one X gene, if they have that trait, they get it. Females, on the other hand, can block it out with their other X, and it's relatively rare, and it's a recessive gene. So they'll block it out with their dominant other gene. And it's the red-green one, like I said, is the most common of color blindness. So, and I can't remember which they call that one, but here's normal vision. Maybe it is this green. This is deuteranopia, protanopia, and tritanopia. And okay, they're just kind of showing what you don't see so well. Because you don't see blue so well, you don't see green so well, you don't see red so well. So there you're showing you what you would see if you had those kinds of color blindness. So, and of course, these are the corresponding with the different cones. All right, that's a lot on color blindness, probably more than you wanted to know. Okay, and ascending pathways, don't need you to know this necessarily either. Go ahead and know that any ascending pathway is going to start with spino and then end with the area that it's going to end up in. So spinal reticular, spinal cortical, spinal thalamic, all these are ascending because, again, they're sensory nerves. Okay, two kinds of localization we can do. One of them is with hearing, okay, and another is with depth perception of sight. So it's called the interaural delay or gap, and that's how we pick up sound of location. So if we hear a certain sound and the gap between that sound hitting our ears tells us where that location is. And have you ever had it where say a siren is going out there and you're looking for the fire truck or the ambulance and all that, and you can't tell because the sound's coming all around you, so it's not able to be perceived. But if it was close to you, of course, you could pick out where it is. But that's a type of sound location we have in it. Again, it deals with the discrepancy between our two ears. And the vestibulo-ocular reflex, and we'll get into that too. But again, you can actually keep your head steady on an image even while your head's moving because of this vestibulo-ocular reflex we have. And I won't go into it more than that, but just kind of know that's a reflex for our vision to be steadied when our head's moving. Okay, this is the one I was telling you about with depth perception, and it's pretty interesting. And so I always have my students take your two fingers basically at arm's length and then extend one just barely beyond the other. And again, with both eyes open. Now try with the other side. Okay, it shouldn't be that hard. Now close one eye. Doesn't matter so much which one. And now try it and see if you can see it. And of course, if you're even in lower light, it's even harder. But yeah, you probably wouldn't be able to see it. Well, you know which one's further and forward or not. But if someone else did it, for example, you may not be able to tell which one was closer because you lost your depth perception. So we need binocular vision for depth perception, and that was known as the retinal disparity between those. This is how we can perceive our depth. And interestingly, animals that are prey animals need that binocular vision to be able to attack the animal. Okay, prey animals would need more around the edges to look for the predators. So it's kind of funny how vision fits the animal's needs, of course. Okay, this is a sensory homunculus. And basically what it is it's a cortical region close to the center of our brain, the somatosensory cortex in this case. And it's literally a map of different parts of our body. And if you sense the sensation in a part of your body, it would show up in that part of the cortex. But as you can see, hands and face are by far the vast majority of it. So if you just looked at your hands and face next to each other, that would be about half of the total cortical area. And of course, we're so sensitive to all those, in those areas, tongue, teeth, lips. That's why it's the central areas, so to speak. Okay, let's keep going. And this was kind of an interesting, whatever concept, but if we had input from the optical lobe, now we're going to relate it to something in the parietal lobe. So they're calling this, a dorsal visual stream. This is the portion that says where it is. Where is that thing? And then if you're going around the corner here down the temporal, this is the ventral visual stream, and this proportion tells you what it is. So yeah, where is it and what is it, and depending on which visual stream you're using, and that would be located in the temporal lobe. All right, we have also a corticospinal tract, and that's just one of these tracts, but that's where most of the signals for a lot of the special senses are using. I don't need you to know this particular fact, though. And that would be the end of that presentation. Kind of fast, I know, but it was very concise too.